Hello friends, we continue our online lessons with new video. Cell activities. Cells, whether plant or animal, achieve many activities. As you remember, we studied and we saw that many of organs which perform different activities are common for plant and animal cells. These activities are reflected on human being because human being depend on its environment, and these activities can be summarized as follows. Simply, in cell activities, we're going to study how these parts of cell perform their functions. Exchange of materials through membranes. Exchanging of material inside and outside the cell is considered the basic cell operation, and we remember it's a function or main function of plasma membrane. This operation regulates cellular functions. Like ribosome depend on amino acids come from environment and determines the exit of waste materials and the water outside the cell. As we studied, we said freshwater organisms need an osmotic balance and discharge out excess amount of water, or by different ways they may have to store wastes in their vacuole like in plant cell, or discharge wastes in different ways in animal cells. For that organs to perform their function properly, it is essential to maintain biological operations of the cell and construction of living matter. Like mitochondria need glucose and oxygen for its operations, and also need to remove CO2 from the cell again through this plasma membrane for its function, or ribosome need different material for construction of proteins. Passage of material is done in many ways. The first one, diffusion. Diffusion is defined as movement of ions and particles in a certain medium from high concentration regions to low concentration regions. Important in diffusion, always particles or materials are moved from high concentration to low concentration. So diffusion can happen in living or non-living conditions without using energy. As a rule, gases like O2 and CO2, because only 2 atoms, CO2 only 3 atoms, they are too small and they can freely pass through plasma membrane. Materials soluble in fats like hydrocarbon and alcohols also are that soluble can spread through biomembranes freely. Freely means without using energy. We have studied that plasma membrane made up of phospholipids, so materials soluble in fats like alcohols and hydrocarbons, they can pass freely through membrane. This can be observed by naked eye when copper sulfide or potassium permanganate are placed in water, which we can see in this picture for copper sulfide. When we draw a part of copper sulfide, all the particles are found together, but as time passes, they distribute different part of glass. At the end, we see a homogeneous distribution of this copper sulfide in water. This copper sulfide and potassium permanganate, they are example for a material which their diffusion can be observed easily in water. And we need these 2 names because question can be asked as fill in the blanks for O2, CO2, hydrocarbons. There are examples for materials which can pass through plasma membrane easily, but copper sulfide and potassium permanganate example for diffusion to be observed easily. Colored material spread on a short distance and it hard spread for a long distance. Distance of spreading particles directly proportional to square root of time means as time passes, particles move away from their source with diffusion from high concentrated medium to low concentrated medium without using any energy. With passage of time, matter will separate all of the water, as we see in this example, diffusion of copper sulfide. Permeability is like an ability of a membrane which allows substance to pass through them or no. So it's an exchange of material between the cell and its environment by aid of plasma membrane. So permeability is an ability of membranes to allow material for passage. The cell can be absorb food material in appropriate food medium. This does not mean that food outside the cell can all be absorbed by the cells as it's needed. These materials must pass through plasma membrane. In addition, this food material should have certain solubility in water to pass this membrane. As we know, when we take a piece of rubber or plastic or glass, because not dissolved in our mouth, we cannot taste it. But a piece of sugar, only we can taste it because it passed through membranes and cell or receptors can detect them. It means any material to pass through plasma membrane need what? A certain level of solubility. Similarly, excess material and feces should dissolve in cytoplasm to pass through the membrane out of cell. As we remember from cytoplasm, about 80% of cytoplasm is water, and also outside the cell means intercellular spaces between cells also contain amount of water. So any material to pass through plasma membrane or a living membrane, they need a certain level of solubility. Generally, membranes are classified according to permeability. Permeable membranes, these membranes allow passage of material regardless of their structure and size. Membranes like cell wall, because their holes or pores are big, so big or small, charged or uncharged material in lipid structure or in different structure, small in size or bigger in size, cast through this membrane, so we call them permeable or even totally permeable membrane. An example we can give as cell wall. Semi-permeable membranes, these membranes do not allow equal passage of solute and solvents. Like plasma membrane or any living membrane, if there's a sugar solution or salt solution, always amount of water pass through membranes more than salt, more than ions, or more than molecules of sugar. Semi-permeable membrane, so we can give as for example plasma membrane as example for that kind of membranes. Selectively permeable membranes, these membranes allow selective passage of materials according to the size of their particles. We have studied nuclear membrane. We see also there are holes, but we call them micropores, which are smaller than pores in plasma membrane. So any material bigger than these pores cannot pass, but smaller or enough small in size, they can pass through these holes. So selectively permeable membrane, we can give example as nuclear membrane. But if for semi-permeable membrane, also we can give nuclear membrane or selectively permeable membrane, plasma membrane, because in structure they are both similar, but only the pores in nuclear membrane are smaller than the pores in plasma membrane. But even plasma membrane selectively permeable, any material bigger than size of pore in plasma membrane also cannot pass through it. Non-permeable membranes like nylon, because there are no pores, no holes or carrier proteins, so no material can pass through it, so we call them non-permeable. It must be noted that permeability of plasma membrane is affected by internal and external factors like temperature, pH, solubility, or charged materials. They all affect on passage of material or permeability of plasma membrane. Osmosis, it is defined as movement of water particles through selectively permeable membrane, like plasma membrane, according to variance in concentration. Means again, like diffusion from high concentrated medium to low concentrated medium. Movement of water particle is done according to diffusion law. In diffusion law, we have seen that materials, particles or ions move from high concentrated medium to low. But for osmosis, material which pass through the or move from high concentration to low have to be water. And it need to pass through a selectively permeable membrane. And if it's water but through different structures, it's not osmosis, or material which is passive, not water, also it's not osmosis. So two rules: the material must be water and have to pass through a selectively permeable membrane to be called as osmosis. But osmosis is a case of diffusion. It works by the same law, which passage of material from high concentration to low. To clearly illustrate the osmosis, we can do a simple experiment using a selectively permeable membrane like cellophane attached to end of the funnel. Funnel, we cover it by a membrane, cellophane membrane, which is obtained from part of animals, which is a living thing, so it must be a living membrane. The funnel is filled with a distilled water until this level. Water, pure water. Then place it in a glass basin. Water level should be the same in the basin and the funnel. So if we place it in a glass basin, but water level going to be same in both of them, because in funnel and in basin, both of them contain distilled water, means pure water, 100%. When sugar is added to the solution, some sugar added, solution added to the funnel, water level rises in the funnel to that level. So there's a difference. Water level rises in the funnel. It indicates that water passes cellophane membrane to the sugar solution. So when we add some solution, before in glass basin, percentage of water 100. It was in fuel 102. But as we add sugar, the percentage of water decrease to about 90. And as osmosis, we said water moves from high concentration to low through a membrane. It indicates that water passes cellophane through solution in the funnel, causing hydrostatic pressure. Water, when passed through membranes, it makes pressure on that membrane, and this pressure of water, we call it hydrostatic pressure. Water particles stop when hydrostatic pressure becomes equal to osmotic pressure. There's an inner pressure of water on membrane and one pressure from environment. When these two pressures are same, water passage to in and out stop. So water particles stop when hydrostatic pressure become equal to osmotic pressure. Solutions can be classified according to osmotic concentration in 3 types. Each type has particular effect in cell. Now we will study. Isotonic solution, hypotonic solution, and hypertonic solution. In isotonic solution, whereby concentration of water outside the cell is equal to concentration of cell cytoplasm, the cell neither loses or gains water. So we remember in cytoplasm as about 80% water. But if in glass basin or in medium also water concentration about 80. So water particles stop to pass through plasma membrane. So cell size not get smaller nor too bigger. In hypotonic solution, this solution has low concentration of non-permeable solutes compared to solutes in cytoplasm. So means in cytoplasm of cell, dissolved material concentration high, but water concentration low. If we focus on water, 70% H2O, but in medium about 80% H2O. So water move from high concentration to low. This solution look and the cell gains water. As water passes into the cytoplasm, this leads to distension of cell and then rupture. And cell how it get water if there is no contractile vacuole, which we study in fresh water organisms. Cell swell, increase until a limited size and then rupture. But this rupturing for animal cell, not for plant cell, because plant cells are covered by cell wall. In hypertonic solution, this solution has high concentration of solute materials compared to cytoplasm. That time, in environment, water concentration is low, for example 70 H2O. But in cytoplasm, about 80% H2O. So water moves from high concentration to low. In that way, thus direction of water is from cytoplasm to the outer solution, to environment. It is caused shrinkage of cell. Cell size gets smaller. But the size of plant cell compared to animal cell does not change when placed into hypertonic solution. Why? Because plants covered by a hard cell wall, the cell membrane inside the cell shrinks, size gets smaller. But hard rigid cell wall, they're not bent, so size in plant cell does not change. But in animal cells, as they lose water, size gets smaller and smaller. The cell membrane merely moves away from the cell wall. We can see only cell membrane move away from cell wall, and this process called plasmolysis, losing water. How we can explain plasmolysis shortly? An animal cell, when we put in a hypertonic solution, water moves from cytoplasm to environment. It loses water, shrinks, gets smaller in size, and we call it plasmolysis. For plant cell, when you put in hypertonic solution, also it loses water, but size not get smaller, because just plasma membrane move away from the cell wall and collapse in the center and get smaller in size. So we call it plasmolysis. It results from water leaving the cell, and this cause of plasmolysis is losing water. When the solution is added to the cell, it goes back to its previous state. It means how in environment there are a lot of dissolved material, we add some material to cytoplasm to balance the concentration, and all this time water moves in. Or we can think like this shrinked cell, we take the cell, we put in hypotonic solution. That time this shrinked cells get water, and at the end it go back to its original size. That time we call it deplasmolysis. This losing water, plasmolysis, gain back this lost water, deplasmolysis. But there are two causes question can be asked from this part. One, Why cell size not get smaller in plant cell during plasmolysis? Because they covered by thick and rigid cell wall. Second, why a plant cell when put in hypotonic solution, it absorb water from environment, get bigger, but not rupture. Animal cell rupture, plant cell does not rupture. So again, about cell wall. Because if you remember function of cell wall, we said it support and protect the cytoplasm and plasma membrane.